Namaskar to all, and you are watching Namaskar Physiology. A successful man is one who can lay a firm foundation with the bricks others have thrown at him. These famous lines were said by David Brinkley. So, determination of reticulocyte count. This is a practical topic. Okay, so we will not be going much into the theory, and thus we will see how we do the determination of reticulocyte count. This practical is done under the following headings: principle, apparatus and reagent, procedure, result, inference, precautions, and lastly discussion. So, principle: a reticulocyte is the immediate precursor of red blood cell and is a non-nucleated cell like red blood cells. Reticulocytes are present in large numbers in bone marrow and only a few, about 1%, are present in peripheral blood. The relative proportion of reticulocytes to that of erythrocytes is determined in a vitally stained blood film which stains the basophilic reticulum of reticulocytes. So this was the principle. So apparatus and reagent. We need glass slides. The glass slides should be very well cleaned and they should be dust free. The edges should be well and good, and there should be not any kind of breakage. Okay, we need anticoagulant such as EDTA, ethylene diamine tetraacetic acid, a compound light microscope, cedarwood oil, incubator, pricking apparatus such as needles or a sterile lancet, cotton, test tube. Okay, and also we need spirit, okay, or antiseptic solutions to to apply on the finger after pricking. So now about the reticulocyte stains. These are very important. The reticulocyte stains are supravital stain. These stains are used for staining unfixed living cells and tissues in vitro, that is outside the body. So here the stress should be given on unfixed and on living. Okay, so there are basically two stains that we deal with: brilliant crescent blue, and the second one is new methylene blue. So firstly, let's see about brilliant crescent blue stain. The composition of brilliant crescent blue stain is, that is, we have to take the brilliant crescent blue. Okay, it stains the RNA of reticulocytes. Sodium citrate, it prevents the coagulation of blood, and sodium chloride, it provides isotonicity. So these are the composition of brilliant crescent blue stain along with the function of that particular component. Okay, now brilliant crescent blue stain is prepared in the following way: that is, 1 gram of the brilliant crescent blue dye is dissolved in 100 ml of citrated saline. That is, citrated saline consists of 1 volume of 3.8% sodium citrate and 4 volumes of normal saline. Here we should know that 1% solution of the brilliant crescent blue dye in methyl alcohol can also be used. Okay. Then nextly, new methylene blue stain. I must mention that new methylene blue stain has a different chemical composition than the methylene blue. Okay, Now the composition of methylene blue stain along with the function of that particular component, these are: number 1, new methylene blue, it stains the RNA of reticulocytes. Sodium oxalate, it prevents coagulation of blood. Then sodium chloride, it provides isotonicity. Now, while methylene blue does not stain the reticulum, that is the basic reticulum, okay, new methylene blue, which is chemically different, stains the material more deeply and uniformly, okay. So new methylene blue is chemically different because it can stain the basophilic reticulum in a very good way, okay. New methylene blue stain is prepared, okay, by dissolving 1 gram of the new methylene blue dye in 100 ml of citrate saline, okay. Now about the procedure. Under all aseptic and antiseptic measures, okay, we have to take around 1 ml of blood from the person, okay, whose reticulocyte count we need to check, okay. Then we can take a syringe of and then we can take 1 ml, okay, and then we have to place the 1 ml of the blood that we have collected in a test tube, and we have to add an anticoagulant such as EDTA, okay, and then in the same test tube we have to add an equal amount of the supravital stain, okay. That is basically we prefer the new methylene blue stain more than that of the brilliant crescent blue, but anyways, so we have to give the supravital stain in equal amount. Suppose if we have taken only 3 drops of blood, then we have to give 3 drops of supravital stain, that is 3 drops of anticoagulated blood with 3 drops of supravital stain. Here, as we have taken 1 ml of anticoagulated blood, so we will take 1 ml of the supravital stain, okay, that is equal amounts should be taken. Now we should gently mix the blood with the supravital stain, and then we need to keep the test tube in an incubator at around 37 degrees centigrade for 15 to 20 minutes. This is to simulate living conditions so that the stain may go to the cell. For suppose if we do not have an incubator, suppose this pen, if you take to be a test tube with the blood inside, then we can do what we can do is that in the palm of the hand we can keep the test tube like this, okay, in a horizontal way, and then we can do like this rotating motions also for like 15 to 20 minutes. We will do like this rotating motion, and then we will do like this, because the normal human body temperature is around 37 degrees centigrade, and when you do that, then you are giving the temperature also, and in the same way you are mixing also. So you can do this, okay. Anyways, after that, the RBCs are then resuspended by gentle thorough mixing. That is, after the incubation is done, we will take out the test tube and then we will mix the RBCs, okay, because they are resuspended. The mixing is also same. We will take the test tube, we will place a test tube in between both of our palms, and then we will mix like this, okay. Now after that, we will make 4 to 5 moderately thick smear of this mixture on glass slides, dry and see under oil immersion. We all know how to make the smears. We will take one glass slide, we will put 1 drop of blood over it, and then we will make a blood film, okay. The blood film preparation is same as that of any blood film preparation, okay. It should have a proper positions, it should be tongue shaped, okay, and the other things as we know to be of an ideal blood film. Anyways, after it is made, we will dry the smear and we will see under the oil immersion, okay, and we will ideally choose the best stain smear for reticulocyte counting. Here we don't have to use any counter stain, okay, we will just see it like that only. Ideally, one more thing, as you can see, this picture is taken from the practical book of that is the Manual of Practical Physiology by Dr. A.K.Jain sir. In this picture, we can clearly see that a piece of paper has been taken. Okay, it has been inserted in the eyepiece. So what is that? Is that ideally one can put a small piece of paper with a hole in its center below the eyepiece lens. This will restrict the microscopic field. It is not a mandatory thing, but you can do it. Okay, I found it in a book, so I mentioned. Use mechanical stage to traverse the full length of the film, and we know how to do that. Okay, now we will use oil immersion objective lens along with cedarwood oil, and then we will bring the blood cells into focus, and then we will identify the reticulocytes. So how will we identify that the particular cell is a reticulocyte? The thing is, the reticulocytes are recognized as non-nucleated cells with the presence of deep blue reticulofilamentous material in their cytoplasm. Okay, non-nucleated cells with the presence of deep blue reticulofilamentous material in their cytoplasm. It is important. That is, deep blue basophilic reticulum with dots, strands and filaments are present in the cytoplasm of the cell. Whereas the RBCs of the red blood cells and other non-reticulated cells are stained with the diffuse shades of pale greenish blue color. Okay. Again, the reticulocytes are also bigger or larger in size. Their diameter is about 8 micrometer or 8 microns than red cells. Now we know that the average diameter of a red blood cell is about 7.2 micrometers or 7.2 microns. Now, for counting, an area of the film should be chosen where the cells are undisturbed and where the staining is good. Now we will count the RBCs in different microscopic fields. We will count at least 1000 RBCs. Okay, at least 1000 RBCs, not less than 1000, but we can go more than 1000. Okay, so we will count at least 1000 red blood cells, and we record the number of reticulocytes encountered in the cell population. We will make a table like this. We'll raise field number of reticulocytes, number of red blood cells. At first field, suppose we saw one reticulocyte only along with say 10 red blood cells, we will write 1 here, 10. In the second one, suppose we saw no reticulocyte and the number of red blood cells is say 20, we will write 20. Okay, these are just arbitrary values also. So when like this is finished by 1000, then we will see that how many reticulocytes are present. It can exceed 10 also. Okay, it's just for reference I am giving. Okay, like that we have to do. The reticulocytes are recognized as non-nucleated cells. I have already told you, with deep blue reticulofilamentous material. Can you see these pictures? These pictures are taken from internet. Okay, these are not my own pictures. I didn't have any pictures, so I had to use it. Anyways, as you can see, this blue color, these are like reticulofilamentous material. These one. Can you see this? These are like RBCs, normal RBCs. These are reticulocytes having deep blue reticulofilamentous material in the cytoplasm. That is, these are deep blue basophilic reticulum is present. Okay, and also their size is also bigger than that of RBCs if you see closely. For convenience, I must say that the reticulocytes are actually precursors of RBC, the just immediate step before RBC is formed. Okay. So these are also some other pictures. This is also taken from net. These are reticulocytes. Okay, these are also reticulocytes. These are normal RBCs. Reticulocytes. This is a good picture. Okay, these are normal RBCs. Okay. So okay, there is one alternative method also. Okay, and in this method we use Leishman stain. So for those who don't know, Leishman stain is used in a practical procedure which is called as differential leukocyte count, or simply DLC. Anyways, we will take here like 2 to 3 clean grease-free glass slides and place a drop of reticulocyte stain in the center of each glass about 1 centimeter from its end. We will, under all aseptic and antiseptic conditions, we will prick one finger, that is the little ring finger for the people who do not know. This is the this is the thumb, index finger, this one. This is the middle finger, this is the ring finger, this is the little finger. Ideally, we use the ring finger, okay, to take the blood samples. Anyways, ultimately, after getting the prick, what we'll do is that we add an equal sized drop of blood to each drop of stain, that is the supravital stain. It can be brilliant crescent blue, 1% brilliant crescent blue, or new methylene blue, okay. Anyways, we will stir with a pin and put the slides on moist filter paper and cover with petri dish. Now we'll allow the mixture to remain on the slides for 1 minute. The slides may be incubated at both temperature for 15 minutes to simulate the living conditions so that the stain may better penetrate the reticulocytes. I must mention here is that both the supravital stains are very good, that is 1% brilliant crescent blue as well as the new methylene blue, both are very good, okay. Generally in our laboratory setup, the 1% brilliant crescent blue is usually available more, okay. I personally have done with the 1% brilliant crescent blue stain, okay. I have done more reticulocyte counts in this stain more than that of new methylene blue. Anyways, and then we will spread a smear of blood, okay. That is, we'll make the smear and we stain it with Leishman stain, okay. Ultimately, the rest of the procedure is same, that is, we will identify the reticulocyte, these will be the non-nucleated cells with deep blue reticulofilamentous material, that is basophilic reticulum with strands, fibrous dots, etc. in the cytoplasm. Also, the diameter of the reticulocyte is around 8 microns, which is more than that of RBCs, which is around 7.2 micrometer. So the size of reticulocyte will also be more than that of RBCs, okay. Well, it is an important note. See, there are two methods. One is direct, one is indirect method. The methods that I just told you, these are what? These are indirect methods, okay. And indirect methods are considered to be more accountable, to be more satisfactory, because the direct method based on the same principle as a erythrocyte count or an RBC counting is not very satisfactory, okay. Simply the indirect methods give more accurate results, okay, than this kind of things. No doubt nowadays we can analyze the reticulocyte count in an analyzer, but if you do manually, then we should prefer the indirect methods. Now, how to calculate the results? See, it's a formula, okay. The percentage of reticulocyte with respect to red cell is calculated, okay. It is calculated by the formula reticulocyte percentage, which is equal to number of reticulocytes divided by total number of RBCs counted multiplied by 100, okay. I've given you an example. See, let the number of reticulocytes counted in a cell population of 1000 RBCs be 15. Suppose during counting 1000 RBCs, we got the number of reticulocytes to be 15, okay. Then according to the formula, number of reticulocyte, that is 15, total number of RBCs counted, that is 1000, okay. 15, 1000, and then this is the 100, this is the 100, okay. This is the 100. Reticulocyte 15, number of RBCs 1000, 100, 100, okay. 15, 1000, 100, okay. So ultimately it will give you 1.5%, okay. Like that we can count, okay. Okay, so now in inference, okay, it is seen whether the reticulocyte count is normal, above normal, or below normal, okay. So we will give the inference, that is whether it is within the normal limits, above the normal limits, or below the normal limits, okay. So that is the whole thing that we need to see. So the precautions, that is what are the things that we need to take care in during and before doing the experiment, and also after doing. And these are the things, that is, the slides should be clean and grease free. There should be no dust. It should be cleaned with soap and water immediately before use. Okay, the glass spreader should be carefully selected, and also the slides should be dry and cleaned. Okay, they should be dried nicely. Okay, it should have a smooth and a clean edge. If a rough edge spreader slide is used, then we know that many of the blood cells accumulate at the edges and in the tail of the smear, and ultimately gross qualitative irregularity in distribution of cells will occur. We will make about 4 to 5 blood films so that at least one may be satisfactory, and we will not heat dry the smear. Okay, stored blood should not be used for making the smear. The blood film should occupy 3/4 of the slide. It should be thin and uniform and should have a fine tail. When looked under the microscope, the red cells should be discrete and not overlapping each other. There should be no Rolu formation on the slide. Okay, Rolu, for those persons who do not know Rolu formation, Rolu, that is the RBCs tend to pile up, okay, with one another, and they form a Rolu. Okay, they accumulate together. So this is Rolu formation, and so there should be no Rolu formation. Otherwise, it will be considered as a bad smear. Now, discussion regarding the reticulocyte count, that is reticulocytes. Normal values in adults and children, the reticulocyte percentage varies from 0.2 to 2%. In average, it is around 1%. Now, absolute reticulocyte count, different books says many things. This is from a book, it says 24,000 to 84,000. Some books say it is between 20,000 to 90,000. Some books say 25,000 to 1 lakh per microliter of blood. So anyways, absolute reticulocyte count is around 24,000 to 84,000 per microliter of blood. Okay, in infants it is more, 2 to 6%. In newborn it is the highest, that is 30 to 50%. It decreases to 1 to 2% in the first week of life. So one hint, suppose if you want to do a practical on reticulocyte count, if you can collect the cord blood, okay, that is the umbilical cord blood, that is if you have a gynecology obstetrics labor room in your department, that is obstetrics and gynecology department in your hospital, and if you can collect from the labor room the cord blood, then it will give you a large number of reticulocytes. It is easier, okay, than that of a regular sample of blood. So it is in very short showing the formation of the erythrocytes. These pictures are taken from Guyton's textbook of physiology. It's a very good book. You can all go through it. This is pluripotent hematopoietic stem cell. Okay, as you can see, it forms many things. Okay, out of them, colony forming unit, spleen it forms, and then ultimately it goes into the colony forming unit blast. Ultimately, it converts to colony forming unit erythrocytes. Okay, now what happens, this converts to pro-erythroblast, then basophil erythroblast, this one, then polychromatophil erythroblast, then orthochromatic erythroblast, then reticulocyte, okay, this one, and finally erythrocytes. I have explained it to you in a very brief way. Okay, that is just lines, but there are many things inside. That is when the hemoglobin forms, what happens, what doesn't happen, this kind of things. But ultimately reticulocyte is formed, and it is just the immediate stage before erythrocytes are formed. Okay, you can go through any standard textbooks if you want to go in details, and it's very interesting also. Okay, and this is very important also. Now, theory of reticulocyte staining. The basophilic remnants of ribonucleic acid or RNA and the ribosomes in the cytoplasm of reticulocytes cannot be stained by the basic dye methylene blue, which is a component of Leishman's stain. The material can only be stained with certain dyes such as brilliant crescent blue or new methylene blue. The dye enters the cells and stains the basophilic material to form bluish precipitate of say dots, short strands and filaments. Okay, these reactions can occur only in supravitally, okay, stained cells, that is in unfixed and living cells. The more the immature cells, greater is the amount of precipitable ribosomal material present in them. Okay, simply the dye, okay, enters the cells and it stains the basophilic reticulum. Okay, and it can do only when the cell is unfixed and it is in living condition. Okay, so these are called as supravital stains. Okay, so it is an exception to reticulocyte because it needs a supravital staining. Now, a few things about absolute reticulocyte count. A direct reticulocyte counting by hemocytometry is not possible, and even and suppose if someone does also, it is not very accurate. Okay, an indirect absolute count can be obtained from the relative percentage by doing a total red cell count. Okay, so total red cell count, it is also a experimental thing. Okay, theoretic, a practical thing. We will not go into details. Okay, if you want to go, you can go through any practical textbook and read it yourself. Okay. Anyways, absolute reticulocyte count, okay, it can be taken out by the following formula, that is, it is obtained by this kind of thing, that is, reticulocyte is multiplied with RBC count, which should be divided by 100. Okay, that is reticulocyte percentage, what you have got, multiplied by RBC count, how much the RBC is. Okay, that is in millions in per cubic milliliter of blood, and ultimately it should be divided by 100. Normal value is around 24,000 to 84,000 per macroliter blood. So some books say it is 20,000 to 90,000. Some books say 25,000 to 1 lakh per microliter of blood, but you can stick to this value. It is around 24,000 to 84,000 per microliter of blood. See, okay, we will come for this for some later on. Now, vital staining. The term vital suggests that this method has to do with living cells. Vital staining is a special method of staining employed for unfixed, that is living cells. Okay, when a cell get fixed, it usually is dead. Okay, so living cells are as nearly as living as possible, including tissue cultures. There are two types of vital staining: supravital staining and intravital staining. So, what is supravital staining? Supravital staining is an in vitro, that is outside the body method, where the living cells are stained by immersing them in a dye solution. The second one is intravital staining. Intravital staining is an in vivo, that is inside the body method, where a dye is injected into a living organism for selective staining. Okay. Now, reticulocytosis. Reticulocytosis means an increase in the number of reticulocytes in the blood. There can be physiological as well as pathological variations. Physiological variations are in newborn infants, we see like reticulocytosis, that is, the blood contains immature red cells, both nucleated and reticulocytes. But after 1 year, there are no nucleated RBCs and not more than 2% reticulocytes. A higher altitude, erythropoiesis is stimulated by the hypoxic stimuli via erythropoietin. Erythropoietin is a hormone, okay, and it stimulates erythropoiesis, that is the formation of RBCs. And we all know that reticulocyte is a stage in erythropoiesis, the stage just before the mature erythrocyte. So definitely it will be more. Okay. In high altitude, what happens is hypoxic means decrease in the amount of oxygen. So this we need oxygen to survive. As oxygen is less, so what happens is that our body tries to produce erythropoietin. Okay, it is stimulated by hypoxic stimulus, and as more RBCs will be produced, we know that the hemoglobin is present in RBCs. So as more RBCs will be there, so the more hemoglobin, and more hemoglobin means more oxygen carrying capacity. Okay, that is more oxygen will be taken. Okay. Anyways, in severe to moderate exercise also, researchers have seen reticulocytosis, as well as in menstruation also sees reticulocytosis. Now, pathological variations, that is after hemorrhage, especially after chronic hemorrhage, hemolytic anemias, okay, basically chronic hemolytic anemias, hereditary spherocytosis, sickle cell anemias, disorders of bone marrow, leukemias, secondary deposits in malignancies, miliary tuberculosis, thalassemia, spleen disorders, also after spleen removal or splenectomy. Also reticulocyte response during treatment of deficiency anemias. We will discuss it. Arsenic and foreign proteins, etc., also produce irregular response without increase in hemoglobin and RBC count. Heavy smoking also induces reticulocytosis in a pathologic manner, according to some studies. Also, any condition which causes the proliferation of bone marrow tissues can cause pathological increase of reticulocytes. Now, reticulocytopenia, that is the decrease in the count, that is reticulocytopenia, sorry, it should be reticulocytopenia, okay? Reticulocytopenia, it is a spelling mistake, reticulocytopenia. Reticulocytopenia means a decrease in the number of reticulocytes in the blood. Reticulocytopenia is seen in aplastic anemia, hypopituitarism, myxedema, this kind of things, okay? There is no documented physiological situation in which their number decreases. Now, reticulocyte response, what is it? Reticulocyte response is an increase in the number of reticulocytes in the circulatory blood. It indicates a high rate of erythropoiesis. Its practical importance is as follows. First of all, it is useful in assessing the erythroid activity of the red bone marrow. Okay. Also, reticulocyte response can be employed to check if the diagnosis and treatment of an anemic patient is proceeding on correct lines. Suppose if an iron deficiency anemia is diagnosed, then we give iron supplements as treatment. Okay, it can be parenteral or tablets. Now, after treating with iron supplement, if it gives a good reticulocyte response, that is, the increase in number of reticulocytes happen, that means the treatment is working, because as reticulocyte is increased, that is, the number of RBC will also increase. So ultimately it is actually a positive feedback, okay, a positive prognostic factor. Also, the potency of a particular drug can also be assessed, say in research purposes, in pharmaceutical researches, okay. The response starts 2 to 8 days after the therapy is started and reaches a peak in the next 8 to 10 days. After that, there is a gradual fall in the response while the RBC count continues to rise steadily. Okay, it needs to point that the reticulocyte response by itself must be interpreted very critically, either as evidence of therapeutic efficacy of a drug or as a prognostic sign or for assessing the bone marrow efficiency. Okay, so one has to be cautious. Note, important note, okay. One treatment is effective, such as for so what is pernicious anemia? Pernicious anemia occurs due to deficiency of vitamin B12, and when we give vitamin B12 injections, okay, folic acid also, then a sudden transient increase in the reticulocyte is seen. This is also called as reticulocyte crisis, okay, because the reticulocyte increase in a sudden huge way. See, many researchers have documented that reticulocyte crisis is a good indicator of effective treatment in case of pernicious anemia. It shows that the patient recovers in case of pernicious anemia. That is, the patient is recovering because and so it is giving a good reticulocyte response. The reticulocytes are increasing. Okay, so it is. Anyways, reticulocyte crisis term is not confined to pernicious anemia's effective treatment only, but it can be confined to anything that shows sudden increase of reticulocytes only. After splenectomy also, we can see the reticulocyte increase in huge number. There is sudden outburst of reticulocytes occur. We can confer the term reticulocyte crisis in that particular aspect also. But reticulocyte crisis that is after splenectomy may not be a good sign, okay. But in case of pernicious anemia, it is a good sign of recovery. So it is a good prognostic factor in pernicious anemia. Now reticulocyte index. The reticulocyte index is a measure of RBC production. The reticulocyte count is corrected for the hematocrit level and for early release of marrow reticulocytes into the circulation, which leads to an increase in the lifespan of the circulating reticulocytes into the circulation, which leads to an increase in the lifespan of the circulating reticulocyte beyond the usual 1 day. Okay, so Ri, that is the reticulocyte index. It is calculated by the following formula. That is the reticulocyte percentage multiplied by hematocrit divided by 45% multiplied by 1 by shift correction factor. Now the shift correction factor varies with the hematocrit. Okay, that is, when hematocrit is 35%, the shift factor 1.5. When hematocrit is 25%, shift factor 2. When hematocrit is 15%, shift factor is 2.5. Okay, like that it varies. So ultimately, if this reticulocyte index is less than 2 to 2.5%, then it implies inadequate RBC production for the particular level of anemia. But if reticulocyte index is more than 2.5%, it implies excessive RBC destruction or RBC loss. Now, some take-home notes about the reticulocytes. The reticulocytes are immature erythrocytes. The reticulocytes are slightly larger, around 8 micrometer in diameter, than totally mature erythrocytes. The reticulocytes are non-nucleated cells. Okay. About 1% average of the red cells in the human body is occupied by reticulocytes. The reticulocytes develop and mature in the red bone marrow and then circulate for about a day in the bloodstream before developing into mature red blood cells. That is also why the reticulocyte response, as I have previously mentioned, the number of reticulocytes increase in the red bone marrow, okay, when it is functioning normally. Anyways, the reticulocytes spent around 2 to 3 days in the bone marrow, and then they came out of the bone marrow and they spent around 1 day in peripheral blood before developing into mature erythrocytes. Okay. The reticulocytes do not have completed production of hemoglobin and consequently contain residual hemoglobin synthetic machinery, okay, mRNA, rRNA, commonly referred to as reticulin. Okay, that is, they don't have like complete production of hemoglobin. The reticulocytes are called as reticulocytes because of a reticular mesh-like network of ribosomal RNA that becomes visible under a microscope with certain stains such as new methylene blue and 1% brilliant crescent blue. That is the supravital stains. So I hope you have understood those aspects. Actually, practical classes, one should show how the practical is done, but I don't have the instruments. Okay, right now I'm in my room, so anyways, I think this should do for now. So these are mangoes in our campus. Thought of giving it. So this is Himanshu Shekhar Gogoi. Thanks and Namaskar. And you are watching Namaskar Physiology. So be safe in this time of COVID-19. Okay, take care of your parents. Be strong. Study well. Stay blessed. And last but not the least, be hopeful. So thank you all.